organization which we'll be discussing. But before that, we must know what an amino acid is. Proteins are large biomolecules comprised of long chains of amino acids. Amino acids are the monomers which combine through peptide linkages to form proteins. They're comprised of two functional groups, amino, which is basic, and carboxyl, which is acidic in nature. There are about 300 amino acids that occur in nature. However, only 20 of them make up proteins. Now we'll discuss the different levels of organization of proteins, the primary, secondary, tertiary, and quaternary structures. The first level of protein folding is the primary structure. This is the sequence of amino acids that make up a protein. By convention, it starts from the amino end and ends in the carboxyl end. This structure is determined through covalent bonds. The secondary structure is where folding starts to happen. The sequence of proteins that we talked about in the primary structure can fold in different ways. The folding is mainly formed through hydrogen bonding. The most common ways of folding are the alpha helix and the beta pleated sheets. Which of these foldings the protein does depends on the amino acid arrangement that it has. The alpha helix is a right-handed spiral structure characterized by hydrogen bonds. It has a N+4 to N hydrogen bonding pattern, which means that the carbonyl oxygen of the nth residue bonds with the amide hydrogen of the N+4th residue. The hydrogen bonds occur every 3.6 amino acids and the pitch of the helix is 5.6 angstrom. Beta pleated sheets consist of individual beta strands connected laterally through hydrogen bonds, forming a pleated sheet. The carboxyl oxygen of one polypeptide chain forms a hydrogen bond with the amide hydrogen of the adjacent chain. Beta sheets come in two varieties: parallel sheets in which the hydrogen bonded chains run in the same direction, and anti-parallel sheets in which the chains run in opposite directions. The tertiary structure is the 3D conformation of the polypeptide. More folding occurs in this shape. A lot of this is dependent on the side chains. When the protein folding is going on, the amino acids with hydrophilic R groups may hang out on the outside, while hydrophobic R groups may be present on the inside part of the protein. The 3D shape is due to other interactions besides hydrophobic interactions too, like van der Waals interactions, ionic bonds, hydrogen bonds, disulfide linkages, etc. There are two main types of tertiary structures: fibrous proteins, which are composed of long and narrow strands and have a structural role, and globular proteins, which have a more compact and spherical shape and have functional roles. Proteins can be made of one or more polypeptide chains, and in the quaternary structure, you're looking at a protein consisting of more than one polypeptide chain. Each of these polypeptide chains can be a subunit, and interactions between them keeps them together. We previously discussed protein folding and the different structures formed after folding. However, this protein may not be ready to carry out its function yet. There might be some additional modifications that need to be made to this protein before it can be functional. Some proteins, however, may also be modified right after translation is completed before the folding occurs. We'll be discussing some of the types of these modifications that proteins undergo so they can carry out their functions. Glycosylation is the adding of a carbohydrate to a protein. This usually happens to proteins that end up being embedded in the cell membrane. This is done because it helps in the identification of different types of cells. Lipidation is pretty similar to glycosylation. It is when we attach a lipid to the protein instead of a carbohydrate. A lot of proteins which are going to be attached to a cell membrane undergo lipidation. The lipid acts as anchors that help to attach proteins to the cell membrane. The addition of methyl group to amino acid chains is known as methylation. It is mediated by methyl transferases and it increases the hydrophobicity of the protein. Many proteins are modified by the addition of a phosphate group. This process is known as phosphorylation. The phosphate group is added to the protein by an enzyme called kinase, which transfers a phosphate from ATP to the target protein. This phosphate group can be removed by a phosphatase enzyme, making phosphorylation a reversible process. It is done to change the 3D conformation of a protein. It can change the 3D structure of the protein and therefore its function. Thank you.